So here's James Watson here at age 22, and Francis Crick. James Watson is actually still alive today, still working in the field of genetics. He just recently took a job this last year in China. Francis Crick is deceased. But in April of 1953, they published a very short 2-page paper in the magazine Nature, explaining the structure of DNA to a T. Again, they did say that they were indebted to Rosalind Franklin and to Morris Wilkins for their knowledge. And this is just kind of a side note. This isn't a Watson and Crick conclusion. But just to give you kind of an appreciation for your DNA, a gram of DNA can store 700 terabytes of data. So it is an incredible information store. But basically, this next picture just shows what they described in their paper. So again, all these measurements that you see are ones that, Rosalind Franklin actually calculated from her photo, and they would have had access to that data. But you'll notice, looking at the, kind of the double helix, it's just not spinning right now. It kind of looks like steps of a ladder. So if we look kind of down the center here, you'll notice that there are 2 hydrogen bonds between the A's and the T's, whereas the G's and the C's have 3 hydrogen bonds. So remember, that's that weak bond that will be broken when the DNA is copying. You'll also notice along the sides that DNA is anti-parallel, which means one strand of the DNA, notice it has a phosphate here. Anytime you see a phosphate, that's attached to that 5' carbon of the pentose, so we call that the 5' end. And then tracing down through that, notice on this end is the 3' carbon of that pentose sugar, and it has a hydroxyl group on it. Okay. If you look across at the other strand of DNA, you'll notice that DNA is anti-parallel. So basically, if I give you a 5' to 3' strand here, then you'll know that this has got to be a 3 and this has to be a 5. So it's basically kind of like two-way traffic. Okay, you'll notice one strand of the DNA had to be flipped upside down to piece it together. Okay. Not too long ago, we actually had an Italian physicist in 2012 that actually directly photographed DNA. Instead of having to crystallize it, he basically kind of looks like a little tightrope here, but he basically kind of suspended that DNA onto these silicon pillars. And so if we zoom in and look a little closer, again, here's that helical structure. Okay, so we've come a long way since Rosalind's photo. One of the things that Watson and Crick did suggest is that DNA replicates semi-conservatively, which basically means, this dark blue represents these 2 parent strands, that those are going to separate or unzip, breaking these hydrogen bonds. And then this old parental strand here serves as what we call a template, a model. So an enzyme can come in, if it sees an A, it puts a T across from it. If it sees a C, it puts a G across from it. And so you'll notice it's semi-conservative because it's still conserving that old parental DNA. And remember, this happens in the S phase of interphase. And DNA copies very, very accurately. Okay. You'll notice there's a lot of characters involved in the steps of replication. So most of these are enzymes. You'll see that kind of by the ASE at the ends of the words. But we're going to basically be looking, and I want you to know the functions of each of these characters. Okay. So helicase is basically going to break those hydrogen bonds. Okay, so that's at the beginning of replication. Helicase would be the first enzyme that comes in and actually unzips that DNA. Okay. Sometimes when you have single-stranded versions of DNA, they form kind of what's called a hairpin structure where they start to kind of fold over on themselves. So to prevent that from happening, we have single-stranded binding proteins that do exactly that. So you'll notice here are some single-stranded binding proteins. Here are some over here. So they basically connect to the single strands of DNA after they've unzipped and prevent them from forming any of those unusual structures. So the name kind of gives that one away. As it unzips, there's a lot of strain and kind of a lot of torque on the DNA molecule. And so there's enzymes called topoisomerases, and they kind of relieve the tension on the DNA. And they do that by breaking DNA at various points, kind of uncoiling it, and then gluing them back together. Then, in order to get replication started, we've got to prime it. And so there is an enzyme called primase, and it lays down an RNA primer. So basically a short strand of RNA nucleotides. Okay, and basically what that will do is it'll insert a free 3' hydroxyl end that's needed to start replication. And so you'll see, primase here, notice it's putting in RNA. Later that RNA will actually be removed and filled back in with DNA. Okay. But if you just think about what you know about primers, you might, before you're going to paint a wall, you might prime it. Before you, put makeup on, you might prime it. I think about like a lawnmower or a snowblower. There's usually a little button where maybe you have to pump that primer before you can actually pull the string and start your mower. So replication requires a primer as well. And then the actual builder, the real workhorse out of all of these enzymes here is DNA polymerase. And so this is the one that's actually going to build the new piece of DNA. And it's really an incredible proofreader as well. So, if it sees an A and goes to put a T in, if it sees a G, it'll put a C, and so on. But if it, for some reason, picks up the wrong nucleotide, it'll basically backspace, delete, and then fix it. And so it's not error proof, but it is an excellent proofreader. And for right now, I want you to write down that this one is the glue. We'll see ligase in quite a few steps of this process. Okay, so notice DNA polymerase this is pretty accurate. In your body, it can actually pick up 50 nucleotides per second, which is pretty phenomenal, but even better is bacteria. Since bacteria do replicate more often and divide, DNA polymerase can actually put in 500 nucleotides per second, but there's a little bit of a trade-off. So the faster it goes, the less accurate it is. But DNA polymerase can only build in this 5' to 3' direction. So for example, okay, here we've had our primer installed, and then DNA polymerase is basically picking up right here where there's a 3' hydroxyl group, and it'll start reading, if there's an A here, it puts a T, and so on, C, G, and it just keeps doing that in this 5' to 3' direction. So we're basically if you kind of look at the new strand being produced, it's being produced in a 5' to 3' direction. Okay, so this end one right here would have another hydroxyl group that could be added to. Okay. The lagging strand actually presents a few issues. So for example, here where we have a primer that has put in RNA, There's that little 3' hydroxyl group. And so a DNA polymerase enzyme can come in and add to that and build again in that 5' to 3' direction. Okay, so that's kind of what's happening here in these 3 pictures. But then notice, as it unzips a little bit more, this end right here is a 5'. We have a phosphate there. We don't have the 3' hydroxyl group that's required to add another nucleotide in. So basically, we'd have to prime it some more. So imagine primase would have to come back in, put this little RNA primer in, 5' to 3'. So it creates that free 3' hydroxyl end. Then DNA polymerase can come in and build up until it butts up against this one. And so this lagging strand synthesis is discontinuous replication, we call it. And it basically builds it in fragments that was discovered by a Japanese scientist. So we can give credit to him and call these Okazaki fragments. And we don't want our DNA to be fragmented. So that's where that glue comes in. So ligase would come in and actually glue these Okazaki fragments together so that they are one continual piece of DNA. Again, the remarkable thing about your genome is that we do have a lot of repair that's available to our cells. So if while it's replicating, there is some type of a mismatch, so if it puts in the wrong nucleotide, DNA polymerase can come in and repair those. So this is the job of DNA polymerase. Okay. And that's going to be basically happening, again, during that S phase of interphase. Okay. But let's say we're in G1 or G2, and our DNA has been struck with, some type of mutagenic agent. So, maybe radiation, certain foods can damage your DNA. There's a long list on the National Cancer Institute of all the different things that have determined to be mutagenic to our DNA. So this is, a lot of different things that it could be here. All tobacco products damage your DNA. So imagine if we're in another part of the cell cycle, the DNA is not in the process of copying, but there is maybe a bulge or a bulky part of the DNA because it's been distorted by some of these damaging chemicals. We have an enzyme that's basically kind of like a DNA scissor. We call those nucleases, but basically they cut out the damaged stretch of the DNA and then replace it with what should be there. Okay, so here's an example. This right here is called a thymine dimer. So we have 2 T's next to each other, but then they notice they form kind of this abnormal bond right here. This is actually caused by tanning beds. So that radiation creates these thymine dimers, and because there's kind of this bulging part of the DNA, nuclease comes in and kind of cuts, makes 2 cuts right here. Another enzyme basically comes in. It'll be a DNA polymerase enzyme to build the DNA. Okay, so it's kind of filling in what should be there. And then lastly, we've got our ligase that comes in and glues it. So excision repair actually takes 3 enzymes. Know that that's nuclease, DNA polymerase, and ligase. And remember back from our cell cycle chapter, we have those proto-oncogenes and tumor suppressor genes. Those tumor suppressors are those guardian angel genes. So they're the ones that actually, kind of screen the DNA, look for things like these thymine dimers, and they activate this repair.